Today, I am pleased to have Dr. Zac Freedman present for us today. He is from the University of University of Wisconsin, Madison. He's assistant professor of soil microbiology, and he's from the O.N. Allen Professor. He's going to talk to us about bioenergy crops, Miscanthus, giganteus, and I just love standing next to him because it makes me feel really small. He's going to talk about how it acts as an ecosystem and engineered to increase bacterial diversity in soil organic matter on marginal land. That's quite a lot to listen to and learn about. So I'm going to turn it over. I'm going to stop share and I'm going to turn it over to Zac.
Well, so first of all, good afternoon, everybody. Thank you for the invitation to speak in this exciting seminar series. My name is Zac Freedman. I'm still feel new, but I'm in my 4th year here at UW-Madison. Before joining the faculty at UW-Madison, I was faculty also for 4 years at West Virginia University in Morgantown, West Virginia. And the story that I'm going to share today was born from my time at West Virginia University, but has continued since I made the jump over to UW-Madison. So yeah, I'm going to be talking about some bioenergy work, about how this giant grass, Miscanthus, can act as an ecosystem engineer to increase soil carbon in Appalachian bioenergy agroecosystems. I'd like to start with some acknowledgements, my old crew there from WVU. And especially I want to highlight this now Dr. Jenny Kane. She did all of the work that I'm going to be sharing today. And she was just an absolutely fantastic graduate student, the right person for the right project. Graduate of Coal City High School in southern West Virginia. When she first started in my lab, she was unsuccessful 2 times for an NSF GRFP, where reviewers said that somebody, quite literally, somebody with her academic background could not be successful in research science, which we all know is BS, and could have really floored Jenny's career. But what she did, she cut out those reviews and pasted them to her monitor at her desk and used them to persevere through graduate school. And now she's a USDA postdoctoral fellow continuing on this project under the direction of Dr. Ember Morrissey at West Virginia University. So really a magnificent scientist, magnificent story. Happy to highlight her work here. And the other people on the grant team that funded this work, Dr. Ember Morrissey, Lewis McDonald, and Jeff Skousen, as well as a small army of undergrads that were involved in getting this research in the ground and off the ground. And this research was funded totally by USDA. And so this is a story about marginal land and how to best use marginal land to benefit both the land and humanity. So the United States has just shy of 1 million square kilometers of marginal land. That's about 11% of the total land area. And today I'm going to focus on a regional story of the Appalachian region, which mainly like southwestern Pennsylvania into West Virginia, where we see a lot of those deeper red colors that indicate a greater area of marginal land. So this is a region rich in marginal land that I would argue is much, much more marginal than what we call marginal land in the kind of upper Great Lakes region, but can kind of discuss that more later. This is also a story about a particular type of disturbance, surface mining. Surface mining or strip mining, this is a practice that removes soil to access natural resources. And judging from these 2 pictures of the Hobet mine, which at one time was the largest mine east of the Mississippi in West Virginia, and the Black Thunder mine in Wyoming, which is currently the number 2 producing coal mine in the country, the environmental destruction of this practice is obvious. And I'm sure I don't have to convince anybody in this room of that. But in surface mining, the topsoil and some of the bedrock is quite literally shaved off of the land to access the resources. And then through reclamation practices, the soil is brought back on and the land is managed to try and maximize its productivity and value. But that's not an easy task. So for this project, we're trying to argue that bio-based product crop production, and bio-based products includes bioenergy, that this may be an environmentally and economically favorable post-mining land use in the Appalachian region. And we weren't the first ones to come up with this idea, of course, that here we have a figure on the left by a modeling study from Milbrandt and others from 2014. And they modeled where in the US would be best to produce different types of green energy. So if you will allow me to zoom in on the biomass figure there from the lower right, where it shows here the more red colors indicate a greater potential for biomass for bioenergy production, and the more green colors indicate less of a potential. If we focus on the Appalachian region, again, mainly southwestern Pennsylvania into West Virginia, we see a lot of these oranges, red colors, a high potential for biomass for bioenergy production. But this land is tough. The soil is of poor quality for sure. So when we were selecting a model crop to try and enhance this soil, put a fair amount of thought into it. And what we arrived at was this giant grass, Miscanthus x giganteus. It's a sterile hybrid of two different grasses, and it has especially high water and nutrient use efficiency, which makes it just grow like wild. And you see there the student Jenny hugging a clump of Miscanthus. They can be quite large, and this was after 3 years of production. Also, Miscanthus is known to be particularly good at building soil carbon. And that's evidenced by this modeling study by Davis and others that modeled the soil carbon sequestration potential of different cropping systems, including Miscanthus, switchgrass, prairie, and corn. And we see over time this long dashed line indicating Miscanthus growth far outperform these other cropping systems. And this was a big bit of data that kind of supported our decision to try and produce Miscanthus on these reclaimed mine sites. And so for this, we were interested in investigating if Miscanthus can act as an ecosystem engineer. And just to get us all on the same page about the vocabulary term here, ecosystem engineers, they're organisms that directly or indirectly modulate the availability of resources to other species. And they could do so by causing physical changes in either biotic or abiotic materials. So in short, they modify, maintain, or create habitats. And the beaver is the classic ecosystem engineer, just for example. And certainly, like all plants can act as an ecosystem engineer for the soil community beneath them, but to act as such in these particular soils takes a quite amazing plant because of all of the different challenges that are faced by the soil that has been reclaimed from surface mining activities. So as a soil microbiologist, the way I see the world is microbes control everything. But a particular process that we were interested in is illustrated by this cartoon on the left there, where we're interested in asking the question, does Miscanthus production alter plant-soil microbe interactions to ultimately increase soil carbon, and not just any soil carbon, stable soil carbon? So if you allow me to kind of talk about this cartoon for a little bit, we have our plant, roots, microbes, and let's just follow the carbon. So start with the carbon as CO2, gets fixed into the plant through photosynthesis, gets translocated below ground either through litter fall or rhizodeposition from the roots. Generally, we can refer to this as plant carbon inputs. The plant carbon inputs are then used as growth substrates for the microbial community. And the microbial community can decompose and assimilate these plant carbon inputs. And in that process, they can produce some carbon back into the atmosphere as CO2 through respiration, or assimilate that carbon into building new biomass. And ultimately, this microbial processing of plant carbon inputs can result in stable soil carbon. A metric that we were particularly interested in is called microbial carbon use efficiency. And on top of this gray box here, to define what microbial carbon use efficiency is, it's the ratio of carbon allocated to microbial biomass over the amount of carbon consumed, where the amount of carbon consumed equals the amount of carbon lost in respiration plus the amount of carbon allocated to new biomass. We measured carbon use efficiency in this system was by tracing the fate of C13 labeled glucose in a jar experiment with field soil, whereby we put some soil that we got from the field into the jar, added the glucose, and then traced the fate of that glucose either into the headspace as CO2 or as microbial biomass. And carbon use efficiency, it's also been shown to scale with soil carbon at a global scale through recent meta-analysis. So it is a parameter that has been shown to be highly related to the trajectory of soil carbon. So in this study, we had the overall hypotheses that Miscanthus production will enhance soil carbon sequestration and the soil sequestration in and the profitability of Appalachian marginal land. And we addressed three specific hypotheses, one being that soil carbon and microbial carbon use efficiency will increase over time since Miscanthus establishment. Two, that organic amendments would most enhance microbial carbon use efficiency, and that effect would be greatest in the most highly degraded soil. And lastly, that Miscanthus production will lead to ecosystem homogenization across disturbance types. So again, acting as an ecosystem engineer to ultimately kind of enhance the soil. So to address these hypotheses, we first selected 3 locations in northern West Virginia that differ in land use history. So specifically, we selected a low disturbance site, which was unimpacted by mining. We selected a moderate disturbance site, which was adjacent to both surface and underground mining, and it was moderate. We called it moderate due to altered soil hydrology and other soil properties. And then lastly, on the right, we had a high disturbance site that was directly surface mined and unreclaimed. So here, the topsoil in some areas are all 3 inches deep. There were big chunks of coal in the soil itself, very rocky, just an interesting site to work in for sure. I should say all of these sites are owned by West Virginia University. And so on the left here, you see our kind of treatment design. So in each site, we had 4 replicates of where Miscanthus was grown. And then we also had different soil amendment treatments, including a high inorganic treatment, high inorganic fertilization treatment, I should say, a low inorganic fertilization treatment, an organic amendment treatment, whereby the amount of nitrogen we added to the soil was equivalent to the nitrogen added in the low inorganic fertilization treatment, and also an unamended control. And spoiler alert, as others have found in these Miscanthus experiments, that the soil amendments had no effect on growth or any properties that I'm going to share data on in a little bit. So some data now. So first, yield. And all the data I'm going to show you is over the first 3 growing seasons of the experiment. And then I'm also excited to report, see a citation on the lower left, that this paper was recently accepted in soil biology and biochemistry, and the PDF went live like last week. So please, if you're interested, check it out. So first, about Miscanthus yield, that one, it increases over time and is lower in the most disturbed site. So we see 2020 is year 2 and 2021 is year 3. We see a statistically significant change in yield across disturbance sites from year 2 to year 3, and these different circles with different colors indicate the different disturbance levels. And I should say the different letters for the rest of the talk indicate different statistical significance. And so we see no difference in yield in either 2020 or 2021 between the low and moderate disturbance site. And in both years, the high disturbance site exhibited the lowest yield. Now some soil carbon data, and across 3 growing seasons here. There was an overall year by disturbance interaction, where overall the soil carbon increased. But interestingly, soil carbon did not increase in the most disturbed site, but it started quite high. And you see that C there being in the year 1, year 2, and year 3. So this was unexpected, and what we think it's due to is the coal in the soil. That the coal, we used combustion analysis for total carbon and nitrogen, and coal is carbon. So started quite high, but again, this project is continuing for another 4 years, and we look forward to tracking that carbon over time. Then here's microbial biomass carbon. This is just across years. There was no difference in microbial biomass carbon across the different disturbance levels, hence a different way of showing the data. And this is pretty easy to understand. Microbial biomass carbon every year increased, which is pretty cool. Then we took a little bit deeper of a dive into the microbes. And first, let's look at the relative abundance of arbuscular mycorrhizal fungi, which can associate with Miscanthus giganteus. And here, the data are expressed across years with the same colors indicating the different disturbance levels from green to yellow to red, low, medium to high disturbance. So we see the AMF relative abundance across years increased across all disturbance levels. And here we see the correlation coefficient, that it was strongest in the moderate disturbance site, less strong in the high disturbance site, and least strong in the least disturbed site, which is pretty cool because we would expect in the more highly disturbed sites those plants to invest more of their carbon in their fungal symbionts to kind of reap the benefits of that mutualism. Next, bacterial richness over time, same deal, bacterial richness went up. The correlation coefficient was highest in the moderate disturbance site, and very similar coefficient in the highest and least disturbed site. And lastly, bacterial evenness, same deal, up over time across the 3 growing seasons. And again, the correlation coefficients here a little bit different. It was highest in the highest disturbed site and the low and the medium disturbance site. A little bit less, but still high correlations, about as high as I've ever seen as a soil microbial ecologist. So, you know, the Miscanthus is improving these metrics, these microbial metrics of soil health. Okay, so now to get into this function of microbial carbon use efficiency. Over the 3 years, that microbial carbon use efficiency increases over time and is lower in the moderate and high disturbance sites as compared to the lowest disturbance site. And so these bars up here indicate that overall there was an increase in CUE in year 3, no change overall in years 1 and 2. But then when you look at the particular disturbance levels, we see the carbon use efficiency is highest in the lowest disturbance site, and then less high in the medium disturbance site, and this should be a B, I apologize. The same for the medium and high disturbance site. But then as we get to year 2 and year 3, there's a slight boost in carbon use efficiency in the high disturbance site. So overall, the microbial CUE is lowest in the moderate disturbance site, which was a little bit surprising to us. It makes sense that carbon use efficiency was highest in the least disturbed site, because we suspect that the microbes wouldn't have to spend as much energy on kind of maintenance respiration, maybe stress response energy, that they'd effectively be the happiest, healthiest microbiome, be able to use most of that carbon to build new biomass instead of other functions. But we were interested in ultimately relating this function of microbial carbon use efficiency to both soil carbon and also microbial diversity metrics. So here are results from a regression where we regress soil carbon against microbial carbon use efficiency. Again, the correlation coefficients up here. And we found significant correlations between microbial carbon use efficiency and soil carbon. These correlations were strongest in the moderate disturbance site. Kind of the middle was the low disturbance site, and the correlations were least strong in the high disturbance site. And so, I mentioned before that microbial carbon use efficiency does scale with soil carbon at the global scale in a recent meta-analysis, but we were pretty excited that these relationships also held given the kind of funkiness of the soil that we were working in for this study. Then we regressed microbial carbon use efficiency with the different parameters of microbial diversity that we measured. And we found that microbial CUE correlated with bacterial diversity at all sites. Again, same kind of graph here, the correlations by disturbance level. The strongest correlations here were in the moderate disturbance site, a little bit less so in the high disturbance site, least in the low disturbance site. And interestingly, microbial carbon use efficiency did not correlate with any fungal diversity metrics. So there seems to be a tight correlation between the bacterial diversity metrics and none with the fungal diversity metrics. And so with this graph here, we're trying to understand how Miscanthus can alter the kind of multifunctionality of soil. And the take-home message I'm going to try to argue here is that Miscanthus production homogenizes soil parameters across disturbance levels over time. Okay, so how we developed this NMDS is we created a single matrix of all of these different soil and microbial parameters that you see in the box on the right. We z-score normalized that matrix and then produced an NMDS plot from it. The colors are the same as they've been for the whole talk with disturbance levels, and year is expressed by the different shapes. So if we go from left to right, on the left here we see a bunch of circles. This indicates year 1, 2019. And if we go towards the middle, we see a fair amount of these triangles, which are year 2, 2020. And if we go to the far right, we see a lot of squares. This is year 3, 2021. And as we go from left to right, we also see a decrease in the dispersion of the points from being very dispersed on the left side of the NMDS, more concentrated or less dispersed on the right. And so we took this as some evidence to support this idea that Miscanthus production does indeed homogenize soil properties, acts as an ecosystem engineer in these soils of low agronomic productivity. So we were pretty excited to see that. So now, if you'll allow me to bring it all together, hopefully you didn't get too angry at me for presenting all of these correlations. I'm going to continue it here. I just want to totally establish I understand that correlation does not equal causation, but still to show these relationships exist are pretty cool. So it all starts with Miscanthus growth. So what I like to think that I just showed you all is that Miscanthus growth has positive impacts on both microbial biomass and bacterial diversity. And as evidenced by these other correlations with your plant height on the x-axis and microbial biomass C and diversity on the y. Then that these changes in microbial biomass and bacterial diversity had positive impacts on microbial carbon use efficiency, as evidenced by, again, these correlations, microbial CUE on the y-axis and on the x-axis, bacterial diversity on the left, and microbial biomass C on the right. Then we posit that these changes in microbial carbon use efficiency underlie increases in both soil carbon and soil organic matter content, and as evidenced by these other correlations of soil organic matter and soil carbon on the y-axis and microbial CUE on the x-axis, where microbial CUE accounts for about 37% and 27% of soil carbon and soil organic matter, respectively. So clearly there are other mechanisms at play, but still that this played out, at least in the first 3 years of Miscanthus growth, was pretty exciting to all of us, and certainly look to see what happens in the next 4 years of the project. So to conclude, we found that Miscanthus production facilitated the improvement of soil properties across disturbance levels, and soil amendments did not influence these trends. And ultimately, that Miscanthus production homogenized soil properties across disturbance levels. So you see, this is the graphical abstract for the paper that we just published. So over time, increase in soil organic matter, decrease in disturbance-driven site variation, and an increase in microbial CUE, biomass, and diversity. And so to revisit our hypotheses to conclude here, our first hypothesis that soil C and microbial C use efficiency will increase over time. We think that was supported by our data. The second, that organic amendments would have some benefit, did not pan out, at least in the first 3 years. And last, that Miscanthus production would lead to ecosystem homogenization across disturbance types. We're comfortable that we showed that with the data that I presented today. So that is all I have for all of you. I hope you enjoyed that, and I'd be happy to take any questions that you may have. So thank you.
All right. Thank you, Zach. If you have any questions, please feel free to put them in the chat box. We do have one question from Ren. She asked, do these correlations differ from correlations measured in disturbed areas where things other than Miscanthus have been grown?
That's a good question. So these relationships that we found are certainly not novel in that we've connected these parameters that others have shown are connected in other systems. I haven't looked in other Miscanthus production systems, like in the CABI project, for example. I know they're growing Miscanthus, and that would be a really great thing to do for the future. But I am confident that on these specific soils or on reclaimed mine soils, that this is the first time someone's looked at these microbial parameters in an in-depth way like we have here.
Okay, thank you. Michaela asked, was Miscanthus grazed or otherwise been removed?
Yeah, good question. We don't have any evidence to suggest that deer browsing or something like that has affected the Miscanthus. We did put fencing around the experimental plots that was electrified to try and prevent that. There were a few times when grazing cows would break through, but we didn't ever observe a cow poop or anything in the plots, and again, no observable loss of biomass to grazing.
Right. David asked, did you normalize measured variables to important inputs like rainfall? We are in a drought in eastern Iowa that has dramatically affected the growth.
Yeah, great question. And we did not in this study. I mean, that would be a very good thing to focus on in future work. Luckily, these years of data that we presented, it was in West Virginia. West Virginia is a very rainy place, and there wasn't a severe drought or anything in these first 3 years. But I will say, as part of the next life of this project, there is a drought component to it, building rainout shelters over half of the plots. And so we're interested in adding an additional axis of variation here to see how differences in moisture content affect these different parameters that we've measured.
Thank you. Steve asks, is the microbial community around Miscanthus unique to it or similar to other crops?
I love the question. Check it out. I was prepared for it. So I wasn't. Thank you, Steve. So we looked at the microbiome using 16S and ITS sequencing for bacteria and fungi, and did show what we have here is the response ratio, where the more bluer colors and the deeper bluer colors indicate more of a specificity to Miscanthus than the nearby kind of business as usual plot where just mix cold and warm season grasses. And so there does seem to be a kind of Miscanthus specific microbiome, but this is not something that we have analytically considered very deeply so far, but we have some data with stable isotope probing to try and better understand the microbial story underlying these things that we've shown so far.
All right. Adina asks, can you talk about the concept of ecosystem engineering versus thinking of your observation of a disturbance response? I think of engineering as a resource management and reallocation, and can see that may be happening here, but also feel like this isn't what I typically think of as engineering. Also, I'm surprised to not see amendment impacts. Could this be because of the strong variations between the different soils? We do see impacts of amendments a lot from ends, so this is different.
Yeah, no, thanks for the questions, Adina. I think, in general, we should talk more about this. There's mutual interest for sure. So with the ecosystem engineer question versus thinking about a disturbance response, so the ecosystem engineers, I was thinking or as we were thinking, I should say, with the team, was that given so Miscanthus has this like wild root system, just incredibly robust, very woody roots, and that can really like mine the soil beneath it. So because of the power of Miscanthus and its ability to kind of enhance these soil properties, especially in the most highly disturbed site, that are pretty confident that the plant is engineering its habitat underneath it in the soil. But then certainly as the plant, growing the plant as a disturbance, differing from the business as usual for how a former mine site is reclaimed. That's an interesting take on it. I think I'd have to think on it a little bit more to compare and contrast those two things. But either way, Miscanthus is empowering the community beneath it clearly. That the community beneath it is different than that in a nearby kind of business as usual plot. So it's clearly heavily impacting its soil microbiome. And then your question about amendment impacts. So there was, like, even if there was some wishful thinking, like the eyeball test, if you will, for statistics, like there wasn't even any response that was anywhere close to significant here. So seems like at least in the first 3 years that there's nothing even close to an amendment response, but those amendments are going to continue, and we'll certainly see how that looks in the coming years. Because the lack of response was so consistent across the different soil types, I don't think it's because of the strong difference between the starting soils, but I mean, it was puzzling and it is continuing, so we'll have to see how it looks in future years.
All right, thank you. Zayinger is asking, in general, what is a nutrient requirement NP and K for Miscanthus?
Ooh, good question, and I don't know that off the top of my head. I do know that we used the 19-19-19 NPK fertilizer, and this is what was previously used in Miscanthus systems in Appalachia. And Dr. Jeff Skousen was the person on the project who lended his expertise with those type of decisions. So I'd have to kind of send you his way.
Steve's asking, have you looked at increased N due to the microbial community fixing N? I talked with a grower in England who thought it might be the case, but unproven. He also follows up and asks if you did a soil test for CEC levels changed with Miscanthus, or if it impacted carbon sequestering. Yeah,
good question. I'll start with the first one about the nitrogen fixers. So we did have a greenhouse study with Miscanthus that was published last year from a master's student, Kieran Liseski, who did use stable isotope-enabled techniques to measure nitrogen fixation. We didn't track the 15N into the plant itself, but did track it from the atmosphere into the soil, and found that in the most disturbed site that there was more nitrogen fixation than in the least disturbed site, which is pretty cool, but the data were super messy. But I do know that Jen, during her postdoc, is planning on using similar methods to look at N fixation in the field outside of the greenhouse setting. So stay tuned for that. And then, yeah, we did measure cation exchange capacity. It didn't make it into the paper, which makes me think that there wasn't anything too dramatic going on there. And again, that's not something that I remember off the top of my head, but if I'm happy to dive back into the data to check it out if you're interested. But we did definitely measure it.